All right, guys, as promised, I want to finish up the remainder of chapter 20 for you. So let's go ahead and do that. Okay, so we left off talking about epithelial cells. We were just getting into this concept that epithelial sheets, right, are another form of our tissues, of our tissue types that we are going to talk about. And we were just getting into the fact that there are four major types of cells that come together to create epithelial sheets, right? Our columnar, our cuboidal, our stratified, and our squamous. And again, these are just based on the appearance of them, right? Very simply, very straightforwardly. And that when we think about the arrangement and how they are put together based on their polarization. So again, we have an apical up here surface. I think I have it in my next slide. Yeah, just for reference, we have an apical surface and we have a basal surface, right? A top or a bottom, right? And how do we know where we are? Because basal, meaning bottom, basement is kind of how I think about it, right? Is the anchoring point of which the cell is going to need to anchor itself. And then, of course, we'll talk about some different junctions and the way we hold them together linearly or in a stratified appearance. But again, that polarization, having the cell orient and say, okay, this is the top apical, this is the bottom, the basal, this is where I'm anchoring, that's going to help to determine how the cell internally arranges itself because the apical surface and the basal surface are going to act in different ways by what they're interacting with, right? It's going to help to determine the arrangement and the functionality of what that cell does. We'll look a little bit more at this when we get into the intestinal tract as an example. So, how do we hold then our very thin layer of epithelial sheets collectively together and at this basal membrane, right? So again, how we do this is through what we call cell junctions. So again, cytoskeletal linked junctions are one of the classifications that we're going to use. But again, we bind these things tightly together so that there is movement and strength for functionality, form and function. So when we think about how our cell junctions are going to come together, how we hold our cells together in our epithelial tissues, we can classify them as being held together by tight junctions. Again, through these epithelial link junctions, which we're going to look at a little bit more, I have in parentheses that there are three major types there. And then our third sort of broader classification is our gap junctions. And again, the reason that we have these is because, again, we're making tissues. We're not looking at the cell individually. We're looking at the cell now as part of a system, right? So we're looking at different tissues that eventually may line and wrap things like organs and then become part of systems, right? And that's very important to have these things arranged appropriately so that they can do their function, form, function, right? So the first being our tight junctions. Again, tight junctions are what really help to bind that epithelial together and make it leak proof, right? We don't want things that should be crossing or not crossing appropriately. So tight junctions make the epithelial leak proof and they separate it at both its apical, right, and basolateral membranes. So again, we're basolaterally sealing together, right? And we're all agreeing, okay, we're at the base, we're attached to a lamina, so we need to attach down here collectively together. And we also need to attach up here at the apical surface together. And how we do that is through our tight junctions. And those tight junctions are held together by transmembrane proteins. Again, it would make sense. I need to extend, when we're going out for a handshake, I'm extending my hand attached to my body to you, who's attaching your hand to mine, right? And we're binding together, but there's still an anchoring point from both of our arms to our physical bodies, right? So we have claudins and occludins that are just the set of proteins that allow for tight junctions to adhere together. back to that broader classification of epithelial linked junctions, right? That we can just kind of think through. Really, all of these are epithelial linked, but we can break down our epithelial linked into sort of three major classifications. We can call them adherens junctions, desmosomes, and hemidesmosomes. All of these, just like we were talking about with our connective tissue being mainly made up of 40 different types of collagens, most of what we're talking about here are made up of a family of proteins called cadherins, right? We just talked about our tight junctions being made from occludins and claudins. These three main types of junctions are made mainly from cadherins, right? These are the protein family that we make up our adherens junctions, hemisomes, and hemidesmosomes. So at our adherens junctions, again, each cadherin molecule is going to be tethered inside internally to an actin filament versus at a desmosome, the cadherin, right, that crosses across and again is part of the hearty handshake, is going to be connecting to a keratin filament. So again, adherens, both binding through cadherins, adherens, cadherins, it's kind of like, could we not have picked something a little different? But adherens are coming together at adherin junctions by cadherins binding to actin filaments in each other's cytoskeletal membranes inside of the cytoskeleton, inside of the membrane. And for our desmosomes, again, still cadherins, but they are actually connecting to keratin filaments. This is really important in places like skin, right? This is really important as part of the elasticity of skin. Hemidesmosomes are really just taking and anchoring through those cadherins to the basal lamina. So one side is still sticking up and binding into a keratin filament, but the other side, sort of the half dome shape, is binding in not to another keratin filament, but into the basal membrane, the basal lamina, to anchor at that basal surface, right? Our last one is our gap junctions, and our gap junctions are exactly what they sound like. They are a literal coring out, right, that allows for cytosolic. Again, where am I? Find my map. Cytosol, cytoplasm inside of the cell, that liquidy medium, right? And now what these gap junctions do is they core out through these connections, right? They literally it's like taking two ends of a straw across that membrane and sealing together at the gap junction and allowing for inorganic ions and very small molecules to freely pass between each other's membranes, right? So again, use the terms to your advantage. Gap junction is literally creating a gap, but forming a junction, right? And boring out between these two so that things can be exchanged versus the other ones we have talked about really are just attachment points. It's kind of like Velcro, right? So think of my tight junctions and my adherens junctions, my desmosomes and my hemidesmosomes as Velcro, right? They're holding each other together. My gap junctions are allowing for, through the binding and sealing, exchange of materials, right? So again, these things are allowing for the connection. And again, just a nice little overview here as far as overall kind of highlight of the things we've just talked about. So this leads into the big, broad classification of how did we all get here? Why are we here? How do things form? Why do things form? And why is this important? And how do we sort of reprogram this? How do we think about how cells lay down that programming with inside of the growing structure, right? So, as far as how our organization is laid out, right, we have a very familiar term, stem cells. right? So how do we create tissues? How do we create which cells become what? How do we identify and organize? It is all based on this very, very early population known as stem cells. And we're going to talk about some of the differences between probably the more famous ones that we think of, which are embryonic stem cells. And we're going to talk about some adult stem cells as well. But because all of the things we've been talking about in class, well, all the things you all have been talking about in class, leading to this point of we're taking cells, we've learned how cells have communicated, we've learned how cells are formed, we've learned how cells exchange with the information of the internal, external, how they regulate, right? Now we're putting cells together into different tissue types. How do we maintain those tissue types? How do we make those tissue types? So again, it all comes together when, you know, you're probably wondering how I got here. It all started when my mom met my dad, right? Absolutely. So as fertilized egg and sperm come together, right? Well, I should say, as egg and sperm come together and become fertilized, now we can give rise to every cell and tissue type in the body. And again, those tissues are going to be organized based on the characteristics and the functionality of the directionality of information they receive to say you are going to become this population of cells and you are going to become this population of cells. And even with inside of our tissues, we have different arrangements of different types of cells. I mean, look at skin. Skin is broken down again into the fatty connective tissue of the hypodermis. Then we have the dermal layer. Then we have the loose connective tissue of the dermal layer, and we have the epidermis. So we have connective tissue mixed with some epithelial tissue, right? And so again, all of these needed directionality. They needed to be organized. And how are they organized is based upon the cues that they receive and how they renew are all based upon very different rates by being identified and eventually becoming a specific cell type. So again, organization and overall operational stability of how we turn over cells, we just left mitosis, and how we maintain tissues is really based upon three major principles. One, how do cells talk to each other? How do cells talk to each other internally? How do cells talk to each other externally? And how does the neighboring junctionality of what we just talked about, how is communication occurring? Two, again, how do we adhere? How and what are we basing against? What am I lining? What am I coming together to form? And finally, the overall memory of the cell, the genetic makeup of I am a skin cell, therefore I will always make more skin cells. That's a memory that had to be installed into that cell type as we were going through two becoming four, four becoming eight, right? So how does this happen? Well, again, it starts with our stem cells. So our stem cells, really, these are the proliferating, right? Making more well, I should say, stem cells are the precursor of precursors. They are the things of which, if we think about the early primordial soup of all things of which life came out of, stem cells are the very, very early cells. Again, after we have the formation of our zygote, you become four, four become eight, and we start to lay down, right? What is going to become the internal layers that eventually differentiate and become different tissue types and different organ types. Those stem cells are it. They are the ultimate cell type because they can become anything inside of the body. So our stem cells all the way up here, again, they lead to the directionality of every cell type of the body. They are the golden, ticket of all cell types. They can become anything. But again, they are laying down that information based upon communication, based upon what the environment, right? It is literally the program cell. So our stem cells then are eventually going to lead into the proliferation, making more, proliferating, making more, of eventually creating precursor cells. I'll come back to this one in a second, of making our precursor cells, from which those precursor cells start to receive those cues, start to really say, okay, I'm going to generate tissue that is connective. Okay, so that precursor cell then begins to proliferate and starts to make different connective tissue types, right? And again, with inside of there, it's receiving information. It's saying, I'm going to become the connective tissue of bone. I'm going to become osteoclast, right? So I'm going to make more precursor cells that are osteoclast because I've got to make a lot of bone, right? But in the end, eventually, when I lay down my official final bone cell, they become what is now called terminally differentiated. It's now a bone cell. And this retains the memory. So does this of saying I'm going to become a bone cell. But once I am a version of a bone cell type, that's it. I am terminally differentiated. I will never be anything but a bone cell. And therefore, everything that comes out of me is forever going to be a bone cell. And in this process, as the body is going through creating and laying down first all of the different types of layers, in that process of growth, when we are growing our embryo, right? These, which we're going to talk about in a little bit here, these we call our embryonic stem cells. And what do we see? This pathway is still the same between both our embryonic stem cells and our somatic, and I'll get to those terms. It's still the same pathway. But as we are making eventually precursor cells that become terminally differentiated cells, we're still going to retain a copy. This is what makes stem cells so incredibly unique, whether they are embryonic or somatic. Because again, eventually embryonic stem cells disappear and we purely move into somatic stem cells because, right, once the body is made, we only become and make these. We don't retain embryonic stem cells forever because we don't retain being an embryo forever. But in this process, as the stem cell itself is proliferating and making more stem cells, it's telling some of those cells, yes, go off. You need to become endoderm, or you need to become mesoderm, or you need to become ectoderm. And out of those layers, we're going to differentiate and make tissues and organs and eyeballs and teeth and hair and whatever it may be. In that process, though, as it's dividing and making two new daughter cells, this is very unique. Only place where we see this really is again, we have our stem cells differentiate or going through mitosis, and one of our daughter cells is going to retain being a stem cell, and one of those becomes a precursor cell. Very unique in our in cellular makeup. Again, this is the foundation of why eukaryotic cells are so unique because they can become any cell. They have the arrangement through evolution and the pre-package through that DNA, RNA, protein to literally have the machinery to say, I can make anything based upon this blueprint. And now I'm going to take that blueprint and I'm going to put it into a machine, stem cell, that can make anything based on my blueprint. It's pretty darn cool. So again, once we get to this point where some cells can make any cell type and some can only make some cell type. Again, stem cells, when we are talking about our embryonic stem cells, or we're talking about a version of potentially somatic stem cells, we are talking about the fact that these guys can make anything. My precursor cells are making what is going to become one specific cell type. They're generating what is eventually going to become a terminally differentiated heart cell, right? So again, think anywhere in the body, and maybe now I'm within a specific tissue, or I'm within a specific organ system, or I'm within a specific arrangement, right? I'm within a specific layer. but I'm not able to just make anything anymore. I'm kind of being narrowed down just a little bit. So let's look at some different examples. Inside of the epithelial lining of your gut, you are constantly shedding and replacing, shedding and replacing, shedding and replacing. And what do I mean by that? Well, inside of the intestines, right? Inside, if I take a cross section here, here's a beautiful little crosshair of all, if I go way, way, way down, again, I'm at a very 100 microns, right? I'm looking very, very small. These little crypts and villi here, this little ups and down, these little finger-like grooves that extend out, this is where the food grinds along after it grinds through your stomach. And as it's riding along these very beautiful thin sheets of epithelium, it's allowing for the molecules that have now gotten smaller and smaller and smaller, and we've broken down my giant sandwich that I've ate into, molecules of maybe polysaccharides, right? Or disaccharides. And now I'm trying to reduce them down and reduce them down to eventually creating individual molecules of glucose so that I can package and put that into the bloodstream. And now the glucose can go off, or maybe in a little bit more of a bigger format, disaccharide, trisaccharide. But again, I'm trying to get it from piece of bread and sandwich all the way down to something small enough to cross into all the cells of the body, right? And feed it to my mitochondria or feed it to the pathway of cellular respiration so I can make ATP. And so if I look at these grooves and knowing that I have to replace this, well, let's look a little bit closer here. Inside of here, let me see if I've gone any closer. Nope. Inside of here, if I look inside of one of these little crypts, right? What do I see? I see that at the very top in the blue here, these are the cells that have to be replaced on a very regular basis. These are the ones that are going to shed all of the time. So that means that if I have to replace up here, then I need to make sure I am making more constantly down here. So let's start down here. Down here, what do I have? I have some that are terminally differentiated. Some of my cells that are here are called Panthus cells, and these are terminally differentiated. They're never going to be anything else. These help to anchor, right? And help me to identify you are intestine. This is a crypt in the intestine, and this is where you are, and you need to adhere, right? Just like we talked about, if I'm a specific type of tissue lining, or if I'm a specific type of epithelial lining in my tissues, got to make sure that I'm where I'm supposed to be, right? And so what do we see? We see that some are terminally differentiated and some are dividing stem cells. So from here, inside of your intestine, you have some stem cells. They are somatic adult stem cells. They're not embryonic anymore because you aren't an embryo at this point. Um, assuming that we're looking at an adult, not an embryo at this particular moment. Um, we're looking at stem cells that are going to eventually lead to precursor cells, right? And precursor cells are going to become some different arrangement of some of these eventual non-dividing, terminally differentiated cells that we no longer need, right? So again, these are cells that are going to be terminally differentiated, and these are no longer going to be replaced, right? Or I should say, they're no longer going to become anything else. But because some of these cell types that are in here may need to be made up of different types of epithelial cells, again, we don't know that. So we have some precursor cells that are available to say you can become this or you can become that. And then I have stem cells that are saying, okay, even within the precursor group, I may have to have some different arrangements. But again, all of this, all I'm trying to show you is that having a regular population of cells that are able to renew, precursorily divide, and then terminally differentiately divide. We have lots of steps to ensuring we can renew our tissues, or we can make a whole system. We can make a whole organism. Same thing, just another example, epidermal layer of the skin. stratified, stacked layers, right? Again, inside, what do we see? We see that we have our basal layer. And inside of our basal layer, we've got some cells. Now we're not getting into as depth of detail as we did here, right? By looking at our stem cells and precursors, but it's the same concept. We have some stem cells, we have some precursor cells, and then we have, right, terminally differentiated cells that are no longer going to divide to become anything else but just this version of cell. And again, what do we also know? We know we have to replace because skin's constantly being shed. We are constantly replacing this. The last and final example is blood, right? Blood is a connective tissue, and blood contains lots of circulating cell types. Blood itself is made up of lots of different types of cells, most of which are out there making sure you are healthy. They're fighting foreign invaders for you, right? On top of delivering oxygen and doing what they need to do. So inside of here, now I have a hematopoietic stem cell, blood stem cell. So again, inside of here, I have one that's going to self-renew. So out of every division of my hematopoietic stem cells, one is going to become some version of a self-renewal. And again, I'm skipping these here because I'm not showing you my precursors, but then eventually what do I make? I can make precursors that can become any of these. So all I'm showing you is again, I need to make sure that as I'm laying this down, I can renew my tissue. How do I renew my tissue? I renew it based on the cell type that I need. How do I know what cell type I need? I need to have cells that are on the ready to go to make the different types of cells that make up my tissue. Those are my precursors, right? Precursors because I may have multiple different types with inside of that tissue. And how do I know that I have my precursor cells ready to go? I have stem cells. And again, stem cells that may be with inside of a given tissue for that specific tissue. So our stem cell tissue renewal, specific signals maintain these populations. We even said, I have to have cell communication. Stem cells can be used to repair lost or damaged tissues. We go all the way back, right? But the difference here is inside of our embryonic stem cells, these are the best of the best of the best. These are the things that can become literally anything. These are the golden goose cells because they are the things that lay down every program of every layer, which eventually becomes every tissue, which eventually becomes every cell, these are the most purest of programmable. And again, it's all based on environment and cues and the blueprint. Once you come out, once you are, out on radar human, you are a baby, right? Now we are thinking about our somatic. What again does somatic mean? Somatic is every cell of the body except for sex cells, not X and Y, right? So we are not talking about sperm or egg here. What we found is through research, again, in our early embryonic mouse models, if we looked at their early embryonic stem cells, this is what we found. This is when it became that very questionable thing, right? Because again, we have to go to an embryonic setup. We have to use an embryo to gain access to these. So ES cells, these embryonic stem cells, they can be maintained inside of a culture forever. And they can maintain to become anything forever. So again, ES cells can be maintained indefinitely as what we call pluripotent cells. If they were exposed to whatever we wanted them to make, and again, even my embryonic stem cells are receiving cues from the environment to say, I need to make this or I need to make that. if they're exposed to the appropriate extracellular cues under what they would normally be doing in vivo, in human, or in mouse, in organism, they can literally differentiate into anything. That's very promising when we think about disease treatment. It's very promising when we think about defects or when we think about, kids being born blind or deaf or missing limbs or whatever it may be. This is exciting. This is beautiful of what it can be. It become any. It can fix anything by replacing damaged cells. But again, these are pluripotent cells that are literally ready to go based on the environmental cues. Because that's a no-no, we can't be going and grabbing embryos. What we have actually found is that we can turn to adult, now somatic, right? So now we're talking about every cell inside of the body that is, except for sperm or egg. We actually have found that we can take adult differentiated, mean they are now saying I'm a heart cell or I am a lung cell, right? And we can actually rewrite their programming and reverse them back into a version of stem cell state. We call this induced pluripotent cells because I have to kickstart that pathway. And so again, we can use these cells to study and potentially treat diseases. What do I mean? So let's say that I have an individual that has one aspect of a genetic disease. I can take a skin biopsy of this. Doesn't mean that that's because that's where the disease is. It could be. But I can take a skin biopsy and I can culture these inside of a dish. From there, I can take and introduce, I can induce through the addition of these um, gene transcriptors to convince these skin fibroblast cells to go back into becoming individual specific, meaning they came just for me, right? Induce pluripotent cells. And now it's like I can take a cell that's at this state and I can regress it back into a version of a stem cell and see if I can reprogram it to become healthy or create healthier tissue. So again, there's two pathway routes we can do. We can take these now induced pluripotent cells. I can use to gene target. I can introduce into these induced pluripotent cells the gene targeting therapeutics that I want to have that be put back into the human to treat the disease state. So again, I can put that into my induced pluripotent cells, and then I can give the correct cues and say, okay, I need you to actually become liver cells because that's where the disease is. Now it's my cells are going to become with my new gene induction that is going to modify and target the disease. I can induce those cells to become liver cells, and now I can take and put those healthy new liver repaired cells back into the human. Or I can go back to the beginning, right? I can take my induced specific pluripotent cells. They're waiting to become what they need to become. I can allow these cells to become differentiated into the affected cell type, whatever the cell, right? I can look at this under a microscope and I can say, okay, I know this individual suffers from liver disease. I'm going to actually look at how this cell forms becoming a diseased liver cell by giving it the external cues to become a liver cell, and I'm going to watch it. I'm going to see how does this disease state happen. Then I'm going to use and I'm going to screen and try different drug therapeutics on it in a culture dish and see which one treats it better without actually having to experimentally test on the human. And whatever one reverses that disease or it has best treatment application, now I can give it the correct drug treatment. So again, this is very promising because again, for a long time, it was only that you could use embryonic stem cells. Now we have found that we can bypass that by using adult stem cells. Okay, I'm going to take a pause here and I'm going to make yet a second video for you guys because I'm sure this one's already long and I will finish it up for you.